Ivan and Marcus, thank you for your wonderful presentation. It is really an honor to have you among us. And I will start with Mr. Štrbák. In what ways can Slovakia and the rest of the Europe can be influenced and inspired by University of Bergen and Norway as such in developing nanotechnologies? It has to be green. Yeah.
Just hold it a bit. Okay, thank you.
Or better say, what would be your reflection towards the presentation that we just heard and seen?
It was really excellent presentation because also we are dealing with NMR. We also make some simulations. So I think the opportunity for collaboration is really great in this field. And I'm looking forward to discuss next other topics. So I think it's an excellent opportunity to set up new collaboration in this field. Because we are also dealing with magnetic nanoparticles for biomedical applications, and we need some simulations. We do also magnetic resonance imaging and NMR spectroscopy, so it's a great opportunity to collaborate.
Michaela, if you could reflect on those two presentations, what is the most fascinating part of it for you?
So it is the possibility to identify at the most fundamental level what happens when it comes to interactions with the cellular membrane. And I think that's, in my opinion, the best way to do it, and how we should construct also our experiments when it comes to the biological effects, taking into consideration what happens at physical, chemical level.
Ivan, from your perspective regarding the new materials which are in various ways innovative, how they will influence the cure of the patients?
Influence the
cure
cure
the process of treatment.
Yeah, well, I mean, novel materials, these nanomaterials the process is to design to be more effective, right? To deliver the cargo or the drug to the tissue that it needs it. So the cancer, for instance, cancer tumor, while avoiding off-side effects. So that's what we are aiming to. It is difficult, yeah, because there is not like the cancer molecule that it's only expressed in the tumor. So it's a bit difficult to have this targeted delivery. There is always some sort of side effect, but we're trying to limit that, to limit this off-side effect so that the therapy is not that harmful for the patient overall, but rather targeting, in this case, the tumor or other diseases.
Could we say, and it is the common question for all three of you, can we say is the bio and nanomaterials are now, let's put it in a way, they're in the diapers and the future is in front of us, or have we already gone through some path? So basically, what's the future in a way that it will be influencing the providing of the healthcare even more and more? Whoever can start.
Well, I mean, there are already in the market drugs which have some sort of nanomaterial composition in it, like Abraxane, for instance. It's a protein-based nanoparticle that has been used to more efficiently deliver the chemotherapeutic drug. And like that, I mean, liposomes have been in the market for several years, and they were used recently to deliver these vaccines for COVID-19 prevention, right? So we have already nanomaterials in the market for drug delivery and nanomaterials used in other applications, like for antibacterial purposes, for instance.
Yeah, sure.
Okay. But only a small percentage of currently used drugs are based on nanomaterials because there are very crucial issues with that safety. So maybe that needs to be solved before they mass scale use in healthcare system.
So basically, the danger of breaking the safety is one of the biggest obstacles that we are currently facing in implementing bio and nanomaterials.
Yeah, definitely, because a small historical excursion. When I was finishing my diploma thesis in 2004, 20 years ago, related to use magnetic nanoparticles in combined hypothermia and chemotherapy of BP6 sarcoma, I read one publication when the author proposed that within 20 years we would be using nano-sized robots in our everyday diagnostic and therapy, but it's far, far from reality. Yes, and
how far? So it's 20 years are gone, so 20 more?
I'm not sure. It's difficult to say because it's a very complex problem. There are several very crucial issues like long-term effects, biocompatibility, because they can easily penetrate every cell, so they can interact with cell organs and biological molecules, which can result in inflammation, oxidative stress, and DNA damage. There's also
how can it damage the DNA?
Because they can be modified by different molecules.
But modify or damage?
Damage.
Okay.
Damage, because these nanoparticles are usually modified by different ligands or molecules, so they can interact with the DNA and damage and cause damage to that. So,
Michaela, can we say in which diagnosis nano and biomaterials can help us improve the healthcare the most? Like, is it in pneumology or is it in
radiology?
Everything? Radiology?
Definitely radiology, because they're also now they're using as contrast agents, so they can enhance contrast, especially in MRI, but they are also used in different imaging modalities, but MRI is the primary choice.
So radiology, and what else?
Well, probably like the most common example for all of us are these paper-based diagnoses for COVID again. So you have these lateral flow devices, and they use gold nanoparticles for detection, for instance. So we use them. I mean, they are out of the body, but we use them for diagnosis.
Did the pandemic of COVID-19 escalated the development and the research and also the usage of nano and biomaterials?
Definitely yes, because for example in mRNA vaccines there are lipid nanoparticles which protect mRNA from damage and prolong circulation time, so definitely. And next environment is next field where they are very helpful is I think cancer therapy. They are now using in magnetic hypothermia, for example, where magnetic nanoparticles are heated to generate localized heat and destroy cancer cells and avoid destroying healthy tissue surrounding. So I think it's very helpful.
Is it helpful especially for some specific kinds of cancer or for cancer treatment in general?
It's developed for general cancer, but surely now in this phase it can be helpful only in some specific cancer.
Okay, well, we can move to the part of the personalized medicine. So, bio, as far as I understand from today, bio and nanomaterials will also influence the system how the personalized medicine is being developed and personalized. So, could you tell us a little bit more about this personalized medicine influenced by bio and nanomaterials?
It can be because, of course, at this level, you can identify better. So one example that was already given was this of cancer treatment. We did our own experiments when it comes to oral cancer. We didn't do it on patients. We didn't reach that phase yet, but we did in the lab on in vitro reconstructed oral mucosa level models, which are 3D models, closer to the oral mucosa, real-life type of oral mucosa. And what we saw was that different types of nanoparticles were taken up by these models, and they were taken up more by the oral cancer models than by the normal mucosa models, probably because of the more disorganized type of tissue that oral cancer has. And another interesting aspect that was also touched by Maria earlier was the fact that we saw higher uptake at intermediate concentrations, not at the highest concentration, probably because of agglomeration of nanoparticles. So at higher concentrations, they have a tendency to sit on top of the oral mucosa models. But what I want to say with this is that, for example, for let's say more external type of tumors like oral mucosa that I mentioned, or skin, they can be used first to identify the places where cancer is localized, and then to deliver also in a more localized manner the treatment. Great help in this respect is the fact that you can excise the tumors where they are. The treatment today implies removal of large areas of jaws and bones and oral mucosa in order for the surgeons to be sure that they have a reasonable safety margin and removing the cancer. So that can be personalized, of course, for each patient based on biomarkers and so on. So that would be an example. Yeah, but when it comes to placing the nanomaterials inside the organism, like injecting them, then there we face the problems that were mentioned. When it comes to the COVID vaccine, which is a very good example, of course, there were very small amounts that were injected, and they were tested materials, so we're, let's say, we think we're on the safe side. Yeah, but when it comes to identifying tumors in the whole body, metastases and so on, by injecting, then, yeah.
But I think at this moment it's quite unrealistic to have personalized therapy by using these bio-nanomaterials, because it would be so expensive, because manufacturing so precise size, shape, and surface charge and functionalize them to every person, it would be so expensive to manufacture. So I'm not sure if it's reality now.
Ben, would you like to add something? He would like to add something to it.
Yeah, well, one of the things we're working with this nanodiamond sensing is, or we're trying to bring up point-of-care device, right, for early detection of biomarkers. So we're using this quantum sensing in order to detect low amounts of biomarkers. So we want to generate kind of a microarray device with extremely low sensitivity or high sensitivities, sorry. But yeah, at this moment, this is an expensive task. You know, these nanodiamond plates
But is it getting less expensive throughout the time?
Yeah, well, as more people start to use it, the problem is that now they are not producing large amounts, like these diamond plates or nanodiamond. They are not producing large amounts, and the synthesis methods we have nowadays, they are expensive. But I mean, we need to improve that technology to make it more affordable. Since it is diamond, what we have, we can reuse it. So diamond is chemically inert, so we can wash, clean, reuse, respot, and test it over and over. So this is something we want to do with this nanodiamond sensing, for instance. But this is, again, out of the body, a type of point-of-care device.
So in order to have this technology more accessible and more affordable, it has to be massively produced?
In part, and to make the production more efficient and more reliable.
And what are the key conditions this has to be fulfilled in order this to happen?
Well, we're working on that. Like we are, so we do this chemical vapor deposition to grow the diamond film, right? So, I mean, the people in the group are modeling growth conditions in the reactors to modify or to improve the shape of the reactor, the type of seeding mechanism, to have a control, and again, batch to batch reliability or good reliability in the production of these films. So we're working on that, but at the moment it's
Can we say that which countries are the most successful? It is definitely Norway, but if you take a look on sort of your competitors, which countries are performing pretty well?
Yeah, well, there is a lot of work from Australia in nanodiamonds. We buy nanodiamond substrates from Japan. They are also good making this type of substrates, so yeah.
May I add, regarding general use of nanomaterials, nanoparticles, I think US and China are ahead, because the European Union is very high regulated, which is necessary, I understand, in the healthcare system, but it slows down the approval of such complex system like nanomaterials are, for example.
But in general, can we say that at the morning I had another discussion and I was asking one of my speaker that if the rule is that usually the United States brings the idea, China produces it, and European Union and Europe regulates it. Something like that? It's not too optimistic, right? We're innovative in regulations.
Yeah.
So what are you going to do with it? All of you.
I'm scientist, I'm not politician, so it depends on politicians.
I would say that we are at the forefront when it comes, but in a good sense. And I think it is necessary, and this discussion shows very well that we have to work together. The developers and the ones that are assessing the hazard related to the use of nanomaterials, assessing the risks, and also what is needed. And a very good contact is needed between producers, scientists, regulators, and of course the public.
And the worldwide cooperation is progressing, but if we could say that which regulations, from your point of view, should maybe be abandoned or modified, or what would help you as a scientist in order, to keep in touch maybe with Japan, Australia, etc., etc. You definitely keep in touch, but maybe to be even further away in the development.
It's difficult for me to speak for the regulators, but from the scientist point of view, what we are already doing and where we are in the front, it is the development of reliable methods and reliable biological models, and bring new test guidelines and standardized methods for assessing the hazard related to nanomaterials, combined with in silico modeling, so that that we can have a reliable evaluation of these materials, and also help the producers, guide them towards, instead of wasting time developing something that proves after many years not to be safe, to tackle these problems from the start. I will say just briefly how it came that we entered nanosafety. We wanted to see if the rejection of implants, especially hip implants, could be caused by nanomaterials, but that was way back in 2004-05. And we have had standardized methods that were going functioning very well. And when we started testing the effects of nano-titania, so titania nanoparticles, we observed that we got, let's say, funny results that couldn't be realistic. So the more we added, the more titania powder we added. We could even see it as a blanket, white blanket in the testing laboratory dishes. The higher the viability of cells we obtained, which of course was resurrecting. That was something completely new. So resurrecting from death the cells. Yeah, so it was obvious that something was not functioning, so then we realized that the nanomaterials were interfering with our testing system. And then we also realized that in order to move forward, we need to have new types of tests, new types of biological models, and that's why we went into label-free testing. And we decided to go into electrical impedance testing, because there you have direct measurements, you don't need reagents, yeah, and the nice effect, let's say side effect but not unwanted, is the fact that they're also ecologically more friendly because you don't need chemical reagents. So, yeah, things are developing on both sides, and we should definitely work together to bring these benefits. But for the time being, of course, the biosensing that Ivan has shown today shows great promise. And it's one area that I see developing. When it comes to dental materials, nanomaterials are incorporated in the matrices of these materials. For example, in polymer-based filling materials, we have nanoparticles or nanofibers incorporated in these matrices, so they are not so easily released. So then we don't face these problems, or at least at a much lower extent.
If you want to add something, feel free to. If we took, I don't know that it's not the most scientific thing to ask, but if you take a look 20 years ago in the future, what are the biggest challenges for you from the scientific point of view in the whole process of developing the new materials?
In developing, because I think
developing or improving or finding out new ones.
If we want to use them in healthcare industry, first of all, we need to solve these crucial questions like long-term effect, because if they are stabilized, they can circulate in bloodstream for a long period of time, and they can accumulate in every tissue, organ, cells, and we do not know exactly how they degrade, what are the metabolic pathways, so what they do when they are in our body for a long period of time. So these have to be solved before it's used in industry, I think, in mass scale. Because now they are using like hypothermia or for like Doxil for cancer treatment, but as I said, it's only a small percentage.
Michaela, from your point of view?
From my point of view, I think we're facing this with every type of new material and everything that we are producing, and gets in there and out there, and then back to us. When it comes to nanomaterials, we have a set of new unknowns compared to previous materials that we have used, that we have to deal with. I don't think it should stop us from developing new technologies that could help us in many ways. It's just to not to be scared, but to proceed with care, and to have a tight multidisciplinary and multi-level collaboration. And if I see some challenges, for me, I see it difficult to address a problem when you don't have a precise terminology or definitions, And I think we have to anchor this, first of all, in the what happens, the phenomena. So, for example, what Marcus has shown us today. So go to the bottom, to the core of the problem, so that we can define better both the materials and terminology related to this, so that when you know what you're dealing with, you know better how to deal with it, how to handle it.
Ivan, what about your perspective?
Yeah, well, I agree with them. It's important to know these long-term effects, or somehow model these possible long-term effects. Well, one additional challenge, at least for scientists, I mean, when you develop an idea and you try to bring that to the market, is extremely expensive, right? So that is a challenge.
Finance is the challenge.
Yeah, that's a challenge, an additional challenge now and in the future, probably.
It will be probably more and more challenging and more expensive.
May I add? Next issue is also ecological issue, like we heard in previous discussion and talk. We do not know how these accumulated nanoparticles will behave in the environment, which effect will they have on the microorganisms or other organisms, so it's a quite big challenge.
We'll see. It will be the challenging times. I hope that the cooperation between Slovakia and Norway, this is just the beginning. So thank you, Mikaela. Thank you, Ivan. Thank you, Oliver. And thank you too.